Hello everybody and welcome back to the JSPS Pathology Grand Rounds, sponsored by the Gene Shanks Foundation and the Pathological Society. I hope you enjoyed your long hot summer, and now autumn is with us. We are back to the Grand Rounds series of lectures. Please can I remind you that if you wish to ask questions, you should write your question in the Q&A box at the bottom of your screen, and we will have a Q&A session towards the end of the lecture where your questions will be read out. So today it gives me great pleasure to welcome Dr. Kerrie Marie. Kerrie completed her PhD in 2013 at the MRC Human Genetics Unit, Edinburgh, supported by the University of Edinburgh, and then in 2014 went on to the National Cancer Institute at the National Institutes of Health to work on mouse models of melanoma metastasis and therapy resistance. Kerrie's research focus is understanding cell fate choices in melanoblasts and melanoma for therapeutic intervention. She has a strong modelling theme using animal models of developmental biology and cancer biology, including use of single cell and computational modelling approaches. Her work is centred around chemical genetics techniques, drug screening and time-lapse confocal imaging of the melanocytic lineage, and she has been working on how the melanocyte lineage pathways are dysregulated in melanoma progression, and she will be speaking to us on this theme this morning. Her lecture is entitled An Embryonic Approach to Uncover New Mechanisms of Melanoma Progression. Thank you, and over to you, Kerrie.
Fantastic, thank you. I'll just share my screen. Can I confirm you can see that okay?
Yes, we can, thank you.
Fantastic. Right, well, it's a delight to be here. So thank you all for having me, and thank you, Sarah, for inviting me. So I don't need to re-say my title because I've already been beautifully introduced. So thank you so much. So I'm a new PI at University of Manchester. I arrived there last year to set up my lab, and I'll explain all about my background in a second. And I'm very, very interested in this sort of cross-section between embryonic gene upregulation and these embryonic genes when they're reactivated in melanoma progression. So just to give you a brief overview of myself, I started my scientific research career at the MRC Human Genetics Unit, mainly studying development, and that was with Liz Patton. And I focused in the fish model for this. So we looked at melanocyte stem cell regeneration and embryonic melanocyte development. And this was sort of an example of some experiments we'd run with chemical genetics and with genetic knockdowns and things like that. And we'd look at, we'd be really interested when the melanocyte stem cells had limited regeneration or when they had enhanced regeneration, and looking at molecular mechanisms to solve this. And what I'd realized is that many of these pathways we were pulling out were actually very relevant to melanoma progression. So I became really interested in melanoma, and particularly melanoma metastasis. So I published a lot with Liz over there. And then I decided to move over to the NCI at the NIH in the USA to work with Glenn Molino. And I really wanted to understand the mouse model, because I wanted to sort of move up in the vertebrate world to have something that was closer, more relevant to humans, but then maybe a broad scope goal in the future might be to be able to combine everything and use an interspecific approach, because every model has their benefits. So I wanted to use this new model and then particularly look at metastasis and melanoma progression. So these were the sort of things I was looking at. I was looking at metastatic progression in the mouse. So, and we'll actually look at these slides in more detail later, and sort of teasing out the molecular mechanisms of these and of therapy resistance, which again was fairly productive. So for any of you that don't know much about melanocytes or melanoma, I'll give a quick intro, but I'm sure you've probably heard a few people from this group talk. So melanocytes make pigments, and they sit at the dermal epidermal junction in the skin. So this is a sort of schematic of a melanocyte sitting next to other cells such as keratinocytes. And in the humans, they're really responsible for protection against harmful UV irradiation. So UV irradiation causes DNA damage. So what the melanocytes do is they release melanin, which can absorb this harmful UV, and they release it in these melanosomes, so these sort of vesicles, and they pass them on to the keratinocytes, and those keratinocytes get to wear them like a little cap to protect their nucleus. And what can happen is when the melanocyte becomes carcinogenic, it becomes melanoma. So melanoma can sort of spontaneously arise, or it can come from moles. People have sort of heard of that one more, but it's not all of the instances of melanoma. And you can have these different growth phases. They can happen in sequence, and they can sort of spontaneously happen as well. But we talk about the radial growth phase being the horizontal growth. So that would be here. And the vertical growth phase is when the melanoma starts to invade vertically down, down through into the dermis, and then eventually reaches the sort of blood vessels and the lymphatic vessels. And then you have different stages of melanoma, like you do with all cancers. So we have regional melanoma stage 3 and distant metastasis stage 4. And why that's important is, well, when the melanoma just occurs in the skin and it's an early stage, you can just cut that thing out. It's easy accessible and you can see it. And so then you have a really good survival rate. But when you start to develop metastasis and the melanoma is now found in lymph nodes, or distant metastasis in organs such as the brain, the lung, the liver, the bone, then your survival rate starts to really plummet. So the metastasis is the thing that's deadly. So one thing that melanoma is known to do is to progress to metastasis very quickly, and that's because, like I said, these patients have poor survival, and it's also known to be very resistant to therapy. Now, traditionally, it was very, very resistant to chemotherapy due to its ability to withstand harmful DNA damage. That's a melanocyte's innate ability. But immunotherapy is doing really good. And in fact, it's one of the poster child cancers for immunotherapy. So if you use these immune checkpoint blockade targeting therapies, around about 50% of patients do really, really well. But over time, around 50% of patients develop either primary or secondary resistance. So we can definitely do better. And what these have in common, metastasis and developing resistance to therapy, is that the cancer is very adaptable. And when I talk about adaptation, I'm thinking about cell state transitions to adapt to maybe new environments or new challenges. So my overarching question is, does melanoma have an innate ability to adapt? Does that come from its parental cell? So we know that melanoma cells, like a few other cancers, are exquisitely plastic, and this is important for therapy resistance and adaptation to a foreign microenvironment. So we know that this sort of cells can transition between different states, sort of given a schematic here, and that this is in response to potential challenges. So my sort of big theory is that cell fate choice underpins this plastic adaptation, and that's what I want to really delve into. So when we talk about cell state transitions and choosing a cell fate, well, that sort of can be a summation of two possible events. One is the cell itself has its own potential that can be encoded epigenetically. So some cells, either stem cells or even cancer cells, can have a potential to become other cell states. And yeah, that's a pre-encoded ability. So sort of schematized here. But then also this potential can be influenced by things that happen in the environment. So the environment will influence the cell and maybe push this cell, which could have been cell A or B, and now it's going to push it into cell fate C, for example. And what I really want to ask is there any overlap between cell fate choice decisions and how the environment influences cell fate choice between the melanocyte in development, so the melanoblast, or between melanoma cells, so the melanocyte in a carcinogenic process, which is in cancer. And I use a developmental lens to uncover novel targetable pathways to control melanoma cell adaptation. And the theory behind this is that cancer is a dysregulated developmental process ultimately. And we all know that cancer is very, very complex. There's lots of different cell types all bunched together in tumours and lots of different things happening with the environment around it. Yet development's very organised, it's very synchronous, we've studied it a lot. So while there's all these interactions here and we don't quite know exactly which one is the most optimal to target, perhaps if we put this developmental lens over the cancer and we understand about these cell-fate transitions, it might enable us to think about a particular transitional potential that we could target that is both in development and cancer. So I think by doing this approach, I'll find some mechanisms of adaptation. Particularly, I'm looking at the ability to switch between cell states. And I think there's advantages to this because, one, if you find something that's exquisitely expressed in embryos, not expressed in a normal adult, but then reactivated in disease progression in cancer, then presumably when you target that, there could be less side effects in the adult, so less secondary effects and unwanted toxicities. The other advantage is that we ultimately, whenever we do our studies, we always have a normal frame of reference with which to understand how cancer is dysregulated. And finally, it gives us a unique perspective. So understanding the parental cells, so the melanocyte lineage. So as I mentioned before, in the embryo, they're called melanoblasts. That's the precursor to melanocytes. And melanocytes can come, they derive in the embryo from the neural crest, which some of you may be familiar with. It's a highly multipotent population of cells. And melanocytes particularly are late migrating neural crest cells. But neural crests can also give rise to many other different cell types, such as neurons, Schwann, glia, bone, etc. So in a way, melanocytes could be considered cousins to these other cell types. And there's particular processes that melanoblasts undergo when they undergo embryonic development, such as, like I mentioned, neural crest cells are highly multipotent. They also have to migrate from the neural tube closure all the way to the skin, which is the largest organ of your body. So they have to migrate extreme distances. And when they get to the skin, they have to cross from the dermis to the epidermis to colonize in the epidermis. And finally, they have to colonize the hair follicle. And in humans, obviously, you have melanocytes both in the hair follicle and in the dermal epidermal junction. In most of our mouse models, they're mainly just only in the hair follicle, unless you use a HGF transgenic mouse. But all these processes I've just told you about could actually have analogous or comparable processes in melanoma progression. So, for example, multipotency potentially could be analogous to plasticity in cancer, and migration and metastasis may share some sort of migratory characteristics. And when I mentioned before that melanoma has to undergo a vertical growth phase to progress and to invade down into the skin, that is almost a reverse of the dermal epidermal migration. And finally, potentially, colonization at their final site in development could have some similar mechanisms of creating a niche that's important to colonization at the metastatic site. And these are the parallels I like to explore in my work. Sorry. So what we know so far about melanocyte development. So there's been a great deal of work by some incredible people. And we know that this is early, the late migrating neural crest cells and there's direct developing melanocytes that travel down the dorsal pathway. So that's sort of schematicized here. So direct developing neural crest cells can become melanocytes. But then there's also this second wave of melanocyte development, which can occur down the ventral route. And that is when these neural crest cells, they sort of cover the peripheral nerves and they form this highly multipotent reservoir of cells called Schwann cell precursors, which are now known to be able to make up many different cell types as a second wave. And we know in melanocytes that this second wave in some models definitely that they can sort of regenerate or develop the majority of the population of melanocytes. So they're super important in melanocyte development. And some of the genes that have been identified that are important in them, for example, are SOX2 to maintain this multipotent state, MITF to cue in a melanocytic lineage. And this is sort of like switching between neuroregulin 1 signaling and MITF. And the cells might be Schwann cell or melanocytes. So that's what we know. And just to give you a final sort of like overview of my hypothesis is that I think these multipotent neural crest cells that are undergoing differentiation and migration to become finally melanocytes in their final area. All these processes are like a reverse of potentially when you have melanoma progression. So melanoma can metastasize and it can also develop plasticity. And I'm looking at the intersection between development and cancer to ask about cell fate choice. So how do I do this? So I've started off using this really exquisite mouse model. So these, we call them the IDCT GFP mice. So they're inducible, you can treat them with doxycycline to induce GFP expression. So essentially this is a tetracycline responsive element. So the RTTA responsive gene is driven by the DCT promoter. So DCT, dopachrome tautomerase, is a melanocyte specification gene that's also involved in pigmentation. And then on the tree element, we have a GFP that's actually H2B GFP. So it's always localized to the nucleus and the chromosome. So it's really bright and good, easy to see. So when we treat with these mice with doxycycline, and in the cells that express DCT, you basically see GFP, which you can see here. This is a little E15.5 mouse. And what you can do is you can also, because they're GFP, you can sort these cells out. So what I do in my preps is I tend to cut off the head and the arms and the legs just to sort of surgically enrich for my melanoblasts. And then I can take out all the inner organs too. And then when I make a single cell suspension of all this, it's like skin, back skin, spinal cord, dorsal root ganglion and things. Then I can actually FACS sort GFP positive cells compared to a tree H2B 1 allele control. And also we can do other things. One thing we noticed when we were FACS sorting these cells is that we would sort of see these two populations of cells. Some had DCT or GFP high and some had GFP low. And that sort of always struck us and we found that interesting. So you can do things like you can just take the back skin of the mice. So if you take a loop of hair and sort of have it on the stick. I actually learned this from Richard Mort back in my PhD days. You can actually use this loop of hair to sort of slowly pick apart the back skin, and you sort of get the epidermis and dermis, and then you can make a single cell suspension of that. And then you'll see that the proportion of GFP high cells is actually enriched, which makes sense because the cells end up in the skin to differentiate. So you would expect that would be where the DCT expression is the highest. And then if we, since it's pathology, I thought we'd show some sections. So then if we sort of look at these mice by pathology, and we go histopathology, and we can section them. So this is a sagittal section from fixed formalin paraffin embedded E15.5 mice. And what I've done here as well is I've stained for anti-GFP. So we then pick up everything that we can see everything that we sort of picked up from the FACS in my studies. And what you can see here is we see really where we expect these melanoblasts to be. We expect them to be in the hair follicle. This is here, but I've kind of flipped it upside down so it's the right way orientated for you. So this is sort of the skin, the epidermis and the dermis. It's sort of quite close together at this stage. This is the outs. This is not mouse. This is nothing. And this is sort of like the adipose tissue and things like that. And we can see the hair follicle is forming here, and we can see these GFP positive melanoblasts are associated with the skin and sort of dropping down into the hair follicle. But we can also see them in other places. So these boxes here are here and here. So this is sort of between, these are peripheral nerves, and then near, this is blood vessels here, and these are the peripheral nerves, and they're sort of between the bones, so between the ribs. And it's one of the places that we could see sort of the melanoblast from some Igor Adameko studies. So I was happy to see them. So we know we're also picking up some of these GFP positive cells that are surrounding nerves. And finally, since we're with the Pathological Society, and I would really love to collaborate more with some pathologists that have an expertise both in development and in cancer and looking at these sections, because I haven't done that extra training, and I know it's a long process and it's a very, very skilled profession. So we have a region here which I find interesting, which maybe is a cranial ganglion, but I'd love someone to jump in and tell me in the questions or something, so which we see a lot of GFP positive in rich cells. So would love to talk to people further about like really dissecting that. So what we can do with all these GFP positive cells that we've collected, and just as a reminder, I cut off the head about here, and I take the arms and legs off when I make my prep and take all of these organs out. So I think single cell sequencing is now not a new process to people. They probably all know what it is. So what we can do is we can take our single cells, and with 10x genomics single cell sequencing, they essentially have in these sort of 10x barcoded gel beads everything you need to do to reverse transcribe and create cDNA from a single cell. So they'll flow them through in one axis, and then you'll start to input cells in another, and the cells bind to these little beads, and they put in oil, so they essentially get closed off in their own single droplets, and you just undergo rounds of heating, etc., to reverse transcribe and create cDNA. So what you get is you get barcoded cDNA when every single cell has its own unique barcode, and you can essentially see the whole RNA transcriptome per cell. And then when you plot them out, it looks like this, lots of colours, lots of different populations. So just to sort of orientate anybody who's not used to seeing these plots. So firstly, I took embryos, should I say, and made my single cell suspension from day 11.5 embryos and day 15.5 embryos. The day 11.5 embryos are enriched for melanoblasts that are crossing from the dermal to epidermal junction, dermis to epidermis, across the junction. And in E15.5, we are enriching for melanoblasts that are starting to colonize at the hair follicle. So then when you see these single cells and you measure the transcriptome, what you can do is sort of do a dimension reduction algorithm and plot them in a 2D space. And essentially every single dot is a representation of a cell, and the closer the dots are to each other, the closer their transcriptomes are mimicking each other, and the further away they are, the more distant their transcriptomes are from each other. And so then you can have an unbiased way to select out these different subpopulations, which we did. And then we took these subpopulations and then we created gene signatures with them. And we didn't just make the gene signatures as were. There was a round of processing, which I worked with my collaborator Vishaka Gopalan from the NCI, where we wanted to exclude genes that were only associated with cell cycle. We wanted to make sure that whenever we were making a gene signature, that it wasn't just that this is cluster versus everything else and what's the most diverse genes. We recognized that we had a few clusters that had common features. So we wanted them to allow to share a portion of these features, but then also find unique parts of the signature too. So just to give you an idea that these are sort of known markers from neural crest development, so we can sort of map them through our clusters and we can sort of see that we're pulling out different populations. So these are your classic, MITF PMEL, these are very differentiated melanoblasts, but you'll have, say, things like ERB3, SOX10, or SOX10 is a neural crest marker, and ERB3 is sort of an NGFR up in Schwann cell precursors, and you'll see them where we expect them to be. So this is all well and good. We're finding new things about the mouse, but does it even relate to melanoma? What's the point? So I believe it does, obviously. But what we wanted to have a look at is just some known developmental markers with clinical significance. So we know from literature that SOX10 low melanoma cells are known to have a matrix remodeling phenotype. We also know that SOX10 high melanoma cells are immune evasive. And we know that AXL high MITF low melanoma cells are resistant to targeted therapy, and that neural growth factor positive melanoma cells can be resistant to immunotherapy. So these are developmental markers that we know we've picked up in melanoma before in the published literature, and they know that they have some kind of clinically relevant features. And what we were happy to see is that we can actually start to pull out different clusters with these markers. So with our melanoblasts, or we could call them DCT-GFP positive neural crest cells, we can see these subpopulations that have very heterogeneous expression, between the subpopulations of these marker genes. So potentially we can find normal cells that are regulating these really important genes for melanoma growth. So we could find out some really key fundamental mechanisms of how these genes and similarly expressed genes are regulated in melanoma. So we have a model. So let's talk about different phenotypic cell states in melanoma and what we know about these developmental genes in melanoma. So this is sort of a schematic, and I've adapted it from a really nice review by Colin Godin and I think Florian Rambo, where they talk about this, they kind of summate everything we know about different lineage markers and different phenotypic cell states in melanoma. And so, as I've sort of mentioned already, MITF can be associated with drug tolerance. So you can have MITF high, SOX10 high cells that are drug tolerant and hyperdifferentiated. If you downshift them MITF a little bit, the melanoma cells become proliferative. You can downregulate that even more and have MITF negative cells that are SOX10 high and NGFR high. They're known to be dormant and drug tolerant. Finally, there's this sort of matrix remodeling feature of melanoma cells where they become axial high and they upregulate this SEB1, which is a mesenchymal transcription factor, and they can be very invasive. And there's sort of like this potentially stressed state that maybe is potentially pluripotent, which I'm quite interested in because maybe it shares some hallmarks with some of our multipotent cells in development. So we can already see some of these features are sort of reminiscent of our development, so maybe they will be good to parallel next to each other. And just to give you a real summation of a lot of work and some key findings, because I'm in the interest of time and I want to get to some other features. When we were looking at our melanoblast data, we overall found that we could predict using lineage tracing that the cells that sort of had a Schwann cell precursor, this sort of multipotent phenotype, were predicted to potentially become these sort of mesenchymal-like GFP positive neurocrest cells. So whether they're still melanoblasts, we're just trying to figure out, or whether we've managed to catch like a cell fate choice where the cell was deciding to become a melanoblast and then switched and became more mesenchyme lineage from the neurocrest. We really need to tease that out. But we could see a subset of these cells in Rachel Bellotti's paper, where she looked at human melanocytes. And we can also see that ZEB1 does stain some VEGF-P positive melanoblasts, particularly those ones that seem to just be adjacent to the peripheral nerve. So without time-lapse imaging, you can't know, but you could hypothesize that these cells might be starting to leave the peripheral nerve and migrate away. So what can we do with all these gene signatures that we've created from development? Well, we created this thing called developmental gene modules, which are, as I was mentioning before, they're essentially this gene signatures which have a few rounds of making them more specific for the lineage, but then also like taking out sort of cell cycle factors too. So we have these gene signatures now, and they're between 50 and 200 genes long. So what we wanted to go ahead and do is basically look in every single data set we could find and classify melanoma and see if we could find anything interesting and if they would sort of stratify or parse out melanoma in a way that was meaningful and maybe would add something to what we already knew. So firstly, you can go to the TCGA, so the Cancer Genome Atlas, and we can look at all cutaneous melanoma samples. Now, these are bulk RNA sequencing samples from the TCGA, so they will be confounded by the fact they have sort of stromal tissue as well as melanoma tissue expressed in them. So you will be picking up signatures for things like T cells and fibroblasts too. But if we just measure the DGM activity in each of these cells, each of these tumors, and then also we were interested at the time in therapy resistance. So it's been published by Peng Zhang that this tumor immune dysfunction and exclusion signature is actually a really good prognostic marker to understand if the T cells are either dysfunctional and not working in tumor cells, or if they're excluded, or you can have neither of the above and they're actually functional, and that's good for the patient prognosis. So we stratified out the patients' samples in this way, and we also stratified them out by their lineage markers, and we just asked simply, is there a correlation? So yes, we can see correlations. We can see that our mesenchymal gene signatures are strongly correlated with tumor immune dysfunction. That's not surprising because already in Begev et al, they published fibroblast high, very fibrotic tumors have poor prognosis and dysfunctional T cells. So that's sort of, we can't tease apart our mesenchymal signatures so well from a fibroblast signature. So that was expected. Also, we repeated probably what people are starting to realize now is that differentiated melanocytic signatures tend to be correlated to good progression, and so they're negatively correlated to tumor immune dysfunction or exclusion. So that's these blue ones. And we also started to see a correlation of our multipotent progenitor signatures with tumor immune exclusion. So this was highly significant, but a slightly weak correlation. So we wanted to delve into it further. And we know that we're confounded with very bulky sort of messy tumors here. So we wanted to look in some immune checkpoint inhibitor treated melanoma cohorts, which are sort of more strictly garnered, and they're sort of they're better controlled, and they're sort of together. It's a nicer cohort, a cleaner cohort to look in. But again, bulk RNA sequencing at this level. And we basically just did a coefficient analysis to see if any of our signatures were correlated with the tumors being responder, so they would respond to therapy, immune checkpoint inhibitor therapy, or non-responders, so they would not. And what we saw is the one standout gene signature was, again, this subset of our Schwann cell precursors, which we call SEP2, and this one was a standout associated with non-responders. So that's led us to be interested in SEP2. So then we ask, can we see any difference in survival? So if you look again this is an amalgamation of multiple immune checkpoint inhibitor studies from the literature, and Vishaka's done a really good job of sort of normalizing them across each other. So we can actually summate them and put them in one data set, so we can have a lot of patients to analyze at once, which is really nice. And you can see that patients that express this Schwann cell precursor signature highly had poorer progression-free survival, and indeed they also had poorer overall survival. So that's sort of making us interested in this potentially multipotent signature, and it's unique from the other multipotent subclusters that we've pulled out as well. So that's quite intriguing and something we're now focusing on. So now we wanted to sort of get a model to really study this and be able to look at progression ourselves, not just by looking at the patient data sets. So I like to use these melanoma cell lines that were derived in melanoma. There's a panel of them, and I'm going to show you stuff from the M4 subline, which is also known as B2905. The joy of these sublines is that they were developed with either just sort of one copy of either an oncogene, or in the case of M4, it was just a HGF transgenic mouse. But they were also developed with an element of somatic mutagenesis. So these ones were UV irradiated. Some of them were DMBA treated. So in this one, we have a HGF transgenic mouse that was UV irradiated, and melanomas developed over a period of 6 months to a year. They were taken out and developed made into this cell line. And we've seen in our studies that they're genetically heterogeneous. So that's more like the patient, that there's lots of different mutations happening at once. And also the joy is that they're made in a mouse, they're syngeneic, therefore you can transplant them back into a mouse that's immune competent. So I can simply do a tail vein injection of these, and when you do a tail vein injection of any cancer cell, they will populate the lung. I like looking at the lung because it's an important metastatic site for melanoma. Ultimately, we could end up doing things like cardiac injections, and they will populate many different sites. But to firstly look at our studies, this is a really nice tractable way where we can get really consistent growth over time. And I'm very interested in longitudinal analysis, so following things 1, 2, 3 weeks. So this is what these melanomas look like. So you can see here a blood vessel, and this is at 1 week when post-injection, and you can see now we have sort of like little micrometastases of this is SOX10 that's staining the nucleus in brown, and you can see here like about 10-15 cells that have been stained there. And then over the course, by 2 weeks, we start to see larger metastases. Some of them start to develop melanin. So we'll see the SOX10 positive nuclei, but sort of this slightly greenier shade of brown that's sort of between cells is melanin. And you can also see large lesions that are unpigmented with some immune infiltrates too. And by 3 weeks, we see sort of very heterogeneous tumours, which we'll look at, I think, in the next slide. So as I've been mentioning, there's some key markers that I've started to pull out my studies, and one big goal of mine is to really take a really big, high, complex panel of those markers and be able to do multiplex immunofluorescence and Hyperion mass cytometry imaging to be able to sort of panel them up in first mouse tissue and then ultimately to optimize for human tissue analysis. So we're just starting off with the basics here. So we have SOX10, which we know is important. It's a melanoma marker. We know it's important in a melanocytic state, and in this Schwann cell precursor state, we always see it being high. We have ZEB1, which we know is upregulated for, it's a transcription factor upregulated with a mesenchymal-like transition. And I always pull out collagen 6A, so I'm kind of interested in that, and I'm keeping an eye on it. But it's, you know, got nothing fundamental yet, but I like to look at it because it gets upregulated just before the cells become mesenchymal-like, and it's sort of in development and in the embryos, it's in these two sort of multipotent state and these mesenchymal cells. So if I stain my tumours, and this is at 3 weeks where they're really heterogeneous, firstly, again, you can see different tumour nodules can have different phenotypes, which I really like because it's a great model for phenotypic transitions and plasticity potentially. So this is a little tumour nodule with SOX10 positive nuclei and actually developing melanin. Anecdotally, they always tend to be softer to like excise. And then this is like a big tumour nodule at the outside of the lung. And this is a melanotic with a few isolated SOX10 positive cells, but not many. And these foci of collagen 6A expression. So these clusters of collagen 6A expression, which is quite remarkable. And if I looked at other tumours and have a look, see what is actually co-localizing with this collagen 6A expression, that they're not co-localized with the SOX10, but they are with the ZEB1, the mesenchymal transition. So we're now sort of really looking at these and studying these over time because they look a little bit like stem cell pools or something. So that's yet to be found out. So again, to sort of bring a lot of long analysis, summatic, because I don't want to go over time. I want to leave time for questions. When we did analysis of our melanoma cells over time, so 1, 2, and 3 weeks post colonization, broadly, if we looked at our developmental gene modules, we could broadly say that early on in colonization, the tumor cells were upregulating these multipotent Schwann cell precursor gene signatures, which sort of was paralleling with what we were seeing with adaptation with therapy resistance. So that sort of piqued our interest. And then over time, the tumours would become more and more mesenchymal-like, so they would upregulate more of those pathways. And what we realised as well is that one cell could actually upregulate multiple of these lineage pathways at once. So we call these the hybrid states. And what we were also realising is over time, the hybrid states were more complex the minute and early on in metastatic colonization. So one single cell would simultaneously express or have an active threshold of many of our different lineages. But over time, we'd start to lose these. And this you can see over here. So the bulk of the cells have like hybrid state phenotypes really early on. And this is from the single cell transcriptome analysis that we did from these melanoma cells. So I took those lungs and I isolated the GFP positive melanoma cells, and then we did single cell RNA sequencing. And then over time that depleted, whereas either our cells with a single lineage sort of phenotype or had no discernible neural crest-like state were increased by 3 weeks. So that led us to believe that this 1 week state or this early metastatic state and adaptation is when lineage features are the most relevant. And so just to sort of like really go into that and understand these hybrid state frequencies, we started to sort of measure these in many different data sets. So you can one cell is considered one of these different hybrid state phenotypes. So you can't have one cell measured in multiple. But so we went and looked at Cell Line Encyclopedia, which is all on a dish and is 2D. And we realized actually most of the cells sit in sort of this classical melanocytic pathways, a little bit of the neural pathways we're picking up. But if you look into patients, things that have been either, we looked through many different data sets, and I'm showing you the human patients, but anything that's basically been an animal or a human is in a complex environment, we would see a massive upregulation of what we call these quad state cells. So these are really highly complex hybrid state cells, which is piquing our interest. Are they more adaptable? Are they more plastic? That's something we're trying to find out. And just to finally to ask if these lineage states can tell us anything new that we wouldn't have just picked up anyway. So we're seeing an evolution over time of hybrid states, which we think are unique and interesting. Then if we pass out, stratify all our cells over 1, 2, and 3 weeks, post-metastatic colonization from the mouse lung that we've done our single cell RNA sequencing on, and we label each cell, whether it's active or not, and we stratify it as one of these hybrid states. So these are sort of depicted at the bottom, the most common ones that we found. And we look at this at 1, 2, and 3 weeks. And what you can see is the overall evolution of main GSEA targets, or GO pathways, I think these are, sorry, is sort of representative of each other, but the actual timing of these evolutions happens differently. So we start to see, for example, these quad state cells don't have interferon gamma first, but they'll have upregulated them by 2 weeks, I think it is. Is it the quad state cells? Sorry, no, it's these tri-state cells. They won't have interferon gamma immediately, but they'll start to upregulate them later. And then we'll have a sort of disparate, they'll all eventually shift to a TNF alpha, NF kappa B signaling. And some of these, it happens at 3 weeks, and some of these, it happens at 2 weeks. So we're sort of seeing a timing difference that we're now teasing out, but a big overall theme is that there's an interferon gamma to TNF alpha shift. And then we're going to now find out if some of these cells are more plastic and if they respond in a different timing to these different potentially environments, or does their lineage allow them to upregulate these signals at a different time. And finally, so we're starting to tease out these pathways that might be overall important, but we think that it's targeting the lineage phenotype is the way we can possibly have a new way of targeting these cells. So how do we do that? So what you can take is from development cells, so we looked in all our development cells and we measured from the gene expression, you can calculate what transcription factors and epigenetic regulators are potentially active in those cells based on what's expressed down. And that's good because in single cell, sometimes you miss like maybe a single gene. So this gives you a really good active signature that's more robust. And we can take those same ones from development now and then ask, in our melanoma cells, in our different stratifications of melanoma cells, are any of these lineage specific, what we call regulons, so something that's a transcription factor, an epigenetic regulator in which we've pieced together from all of this transcriptome. So are any of these regulons still relevant, and are they more active in particular cell states? So if we look at these hybrid versus non-hybrid, we can see that in our mouse melanoma cells, and then we also wanted to see are these human specific. So we could see that regulons that were upregulated in our mouse melanoma cells hybrid states were also upregulated in human cells. So these are potential targets that we can go and inhibit and go look and see if and we hypothesize they'll modulate the phenotype of the cells and their ability potentially to adapt. And this is how we're going about this now. So my program of study consists of 3 specific aims. One, I'm mapping neural crest sulfate decision points in these DCT specified cells in the mouse embryo. 2, I'm establishing the functional importance of these lineage states in early melanoma metastatic adaptation. I have lots of correlative evidence saying that they are important, but now we're going to delve in and we're going to knock out, we're going to interfere, and we're going to see, are they really important? And finally, if that all bears fruit, I'm exploring these sulfate regulators as potential targets for therapy. So I've developed some reporter lines. Right now, there are single reporter lines, and the ultimate goal is to combine them together, where I have M. scarletti on SOX10. So these are CRISPR tagged lines, so it's on the endogenous gene, And what we have is we have a two-way cleavable linker. So whenever you have one copy of SOX10 made, you have one copy of Scarletti, but they're cleaved, so you don't affect the protein, for example. And we can do the same with a different fluorescence for Col6A1 and for ZEB1. So we know SOX10 and ZEB1, for example, show very divergent phenotypes. So this is a really good way to now look at these phenotype cell state transitions in real time. So I've got them separately and eventually we'll combine them together. And what I've been doing is looking at our targets in 2D growth and inhibiting them. And anecdotally, we don't really see much change in 2D growth. But what I'm really interested is when you don't see much change in 2D growth, but then if you put the cells in some kind of increased complexity of 3D cultures, if we actually start to see phenotypes. So I gradually increase the complexity in these in vitro models, so I can use things like hang and drop to create this anchorage independent growth and simulate anoikis and 3D organization. I can also plate these colonies or single cells into matrices to look at how they the cells themselves interact with surrounding matrices, of which these are sort of the most important substrates that I'm sort of keying into based on what's relevant for the lung. And finally, I'll look in vivo in my lung metastatic experimental metastasis models. So over time, if I see phenotypes, I'll be able to sort of pinpoint at what point in the metastatic cascade and the real feature, where is that cell important? Is it adaptation to anoikis? Is it survival at a foreign microenvironment? Or is it the cell's ability to grow in a matrix, for example? And this is just sort of an example of one of these colonies growing in matrices. When we look at them in 2D, they just look the same. They're very, very homogeneous. They do not pigment or anything like that. When you put these cells into 3D matrices, a bit like in the tumor in the lung, you start to see that they have these sort of heterogeneous morphologies. They have these tubular invasive structures. They have proliferation out here. They potentially have these darker cells, which I need to do sectioning to confirm if they're actually producing melanin. And this is Cy3 labeled collagen. The collagen does get reorganized around them. So whether they're actively doing it or passively doing it, I have to find out. And finally, now my lab is growing. I have 3 PhD students just starting, and in collaboration with Sarah Woolner, who is really amazing at mechanical modeling, at stretch and proliferation. And we're going to ask if there's any mechanical influences that mediate cell fate choice with Johan, and that's in development mainly. Charlie's a mathematician, so also working with Oliver Jensen, and we're going to mathematically model some of these cell fate choice decisions. And Yu Hong's come over, and she is working with Sarah, me, and Colin and Ian, and we've got some looking at RAS mutations in melanocyte development and melanoma. So in summary, I believe that there is a mesenchymal-like state in the melanoblast lineage. Whether or not it's we're seeing a difference in cell fate choice and the cells are now specifying to melanocytes and then changing, we're going to find out that there are these hybrid states. I didn't show you today for time, but I can tell you they're rare in development, but they're very enriched in melanoma. And that we see these developmental states are associated with tumor evolution and with therapy response. And now we want to see if they're targetable, and that we do see an inflammatory signaling switch also associated with tumor evolution. And whether or not this is tied to developmental lineage, we will find out. And I've sort of told you most of my compelling research questions already, but I want to look more into these inflammatory signals and these the role of matrix remodeling melanocyte lineage in development and disease. But I also want to understand these hybrid states and the plasticity capacity of them in tumor evolution, and can ultimately we target cell fate choice. So I love being at Manchester. It's such a collaborative environment, and I love being in the Northwest because we just had an amazing meeting last week, and we have great neighbours at Liverpool and Lancaster as well. So I look forward to talking to you all and taking your questions and maybe collaborating in the future. With that, I'd like to thank everyone, especially Vishaka, who's worked with me very closely on the project.
Okay, thank you very much indeed, Kerrie. You can't hear the sound of clapping, but I'm assuming lots of people are clapping vigorously at the moment. So thank you very much indeed. That was a really nice talk, well explained with some beautiful images, some lovely histology and immunostaining. Absolutely fantastic to see that. So whilst we're waiting for people to put some questions in the Q&A box, I'd like to kick off with the first question, if I may.
Yeah.
When you were talking about the mesenchymal-like cells and the plastic cell pool being able to change from one to the other, what do you think is driving that process of change?
So in development or in cancer?
Well, both.
So I sort of quickly highlighted this inflammatory switch, and we do see TNF alpha upregulated in cancer when the cells are, when we study them, we see that they're more mesenchymal. And we kind of did this full circle, learning from development, mapped onto cancer, and then we found that in cancer. So then we kind of mapped back and went, do melanoblast, or do these DCT GFP positive cells ever upregulate inflammatory signals? And we did actually see a positive correlation, significant only with the mesenchymal lineages and one of our multipotent lineages, which we think maps, which we think is more closely related to mesenchymal. So potentially it could be that was my sort of first thing to look into, if it is an inflammatory switch.
Okay, thank you. Right, I'll move to the Q&A box. So the first question is from Stephen Notley, who asks, thanks for a really great talk. Have you studied the role of site-specific (brackets lung) tumour microenvironment in relation to cell state switching in your model? There are recent studies showing that the lung AT2 cells, through reciprocal interactions with tumour cells in breast and colorectal cancer, are essential for this phenotype switching between tumour cells.
Great question. So yeah, it's on the goal of what I want to do. I've so far just been looking at if I can see what we're trying to do is sort of stain for all our lineages, and then we have stromal markers, and we're going to do segmentation analysis to see if anything's closer or not. What we also have plans to do is single cell transcript. So what we didn't do with this transcriptomics in the lung is take much of the stromal tissue. We enrich for GFP positive. So we want to go back in and take both our melanoma cells and our lung microenvironment. And my collaborators have like a really nice sort of derivative of the cell chart algorithm that they've developed that can really tease apart some of these sort of interactions in cell chat between stromal and your melanoma cell, but you need both populations to do that. So it's on the go, and I don't know much about lung AT2 cells, but I'm going to like write that down and have a little look, because maybe they have something. There was also a really nice paper from Chris Marine's lab last year, which showed that the endothelial cells themselves were signaling and allowing a change over time in sort of like a broad neural crest-like phenotype. So there could be signaling pathways either from, the blood vessels themselves, or, I mean, I'm also very interested in what are the nerves doing, because we see that's really prominent in development. So I'll start looking at them. Thank you, Steve.
Okay, so the next question is from James Morris, and he asks, does malignant melanoma have a consistent microbiota? If so, any plans to study this feature?
No plans. Probably, though. I think I didn't go to MRA, but if Liz is online, she might have done. I think Adam told me that there was a really beautiful talk last year or this year about a lady who's studying microbiome in melanoma. That is so beyond my expertise, but I find it fascinating. But no plans. I'm not the right person to do that.
Okay, thank you. I have another question for you. One of the things you talked about that caught my interest was the SCP2 population of cells, comparing SCP2 high with SCP2 low, and you showed that there was a difference in survival, with poor survival in the SCP2 low cells. Why do you think that is?
Yeah, so yeah, we have to find out. So potentially the SCP2 cells are more multipotent, so they have more stem cell features attached to them. The SCP1 they have slightly more inflammatory signals. That's a population do. So maybe at E15.5, there are two subpopulations, and one cell's retaining much more sort of undercover multipotent phenotype, whereas another one is either differentiating and becoming or being stimulated by the inflammatory environment. I always found it wild that it was SCP2, which is a an E15.5 signature versus, I mean, when I went into this, I thought all the early embryo 11.5 will be chock-a-block full of things that are associated, or gene expression that's associated with poor progression, because we always think globally de-differentiation is really important. So it's actually a subset of what we think are maybe more multipotent cells in a later embryo. So yeah, we're going to tease that out, but I think they're more multipotent and potentially they're hiding away from the immune system.
And do you think that might reflect that when the cell has to first establish itself in the very early stages of metastasis, that it requires a range of properties, and so being multipotent and undifferentiated is advantageous, particularly at the early stage?
That was my whole hypothesis going through that. Can we find that out? Because that would, if you sort of think of it logically, would be the most important. It would need to. I sometimes show this figure of Conrad Waddington's landscape, where epigenetically in development you see cell fate decisions being like a ball rolling down the hill, and the ball can only go downhill, and once it's picked a fate, it sticks to it, and that's sort of been around since 1958 or something. But when you think about cancer, you can think that the ball now magically can defy gravity and propel itself back up the hill. So is that ability to be able to sort of reverse and deregulate really important? And, and I think some people have shown that deregulation is important already. And what I want to know is, are they the same pathways? And is there like this hub state that is associated with development, and it's sort of actually an innate ability of the cells that they've always had since they were in embryonic progenitor. But yeah, I think it's the most important time, and it's why I was really excited that our early metastases are enriched for all these hybrid states, because that bears fruit with that hypothesis.
And then once the tumours are established as small groups, small clusters of cells growing in the parenchyma of the lung, presumably they then need to switch their properties to a more growth orientated set of characteristics.
Yeah. So we're going to do cell cycle analysis with these as well. The tumours do bulk up very, very quickly, and we do find the tumours that tend to be unpigmented are thicker. They're more fibrotic. You can sort of, whereas the melanin heavy ones are sort of looser, these ones are thick. You can almost gouge them out quite easily, and they tend to be full of sort of our mesenchymal markers as well. So that's probably associated with like outgrowth and sort of the tumor just sort of going wild, which possibly is shared in a pan-cancer way. So once they hit that growth phase.
Okay, thank you. So just to note that James Morris has come back with a reply on the microbiota. He says microbiota is relevant to inflammation. Does this make it worth thinking more about?
Yeah, I mean, we should talk. If you're a microbiota person, then we should talk, because I would definitely love to chat to people who know more about it. It could be really relevant. You're right. I guess in the embryo, we're talking like about there are no microbiomes in the embryo, so we're sort of, there's a less immune system in the embryo. I mean, at the time we're studying by E15.5, you start to see macrophages start to infiltrate the trunk. So that's sort of the immune system that's around at that time point. Microbiome less so, but definitely in the lung parenchyma, there are going to be microbes, and it could be really important. But yeah, contact me, and then I've got your name as well, and we can talk, and we can see if there's anything we can tease out. Perhaps it's already there in the sequencing, and we can look at that.
Okay, fantastic. James says he works with people in Lancaster.
Okay, brilliant.
Probably not too far away from you in Manchester.
Yeah, not far away, and I work with Lancaster a lot. I talk to Richard Mort a lot, who's in Lancaster as well, so always happy to go through to Lancaster and visit.
Great. On that very positive note, it's 1 minute to 10, so I think it's time for us to stop. We have a tradition of stopping at 10 o'clock exactly and giving people plenty of their morning back. So can I just finish by thanking you again? That was a really fascinating talk. I enjoyed it. I'm sure almost all the audience have really enjoyed it as well. So thank you very much indeed.
Thank you so much for having me. Really appreciate it. And if anyone has pathological expertise and they want to help me look at loads of sections of tumours and embryos, I would love to chat to you. Thank you so much.
Okay, Thank you, everybody, and goodbye.